just basically you can look at the whole playlist of recorded lectures. If you haven't seen it yet, it's just the link in the modules part of Canvas. Up at the top, we'll say YouTube playlist of like recorded lectures or something like that. So you go there and just find the date that you missed. And I think I just looked, some people have watched a few of the lectures for ones that they've missed. So they are accessible and viewable. But I'll also post the Kahoot quiz link and I'll point out where it is on Canvas. So hopefully everyone can get back into it. We'll go over the quiz. We'll do a lot of reviewing on Friday. The quiz you guys took on Friday, I'll do the diffusion worksheet and we'll do the Kahoot review quiz. So hopefully that'll be a really good all-around review for the exam on Monday. Okay, what am I doing now? The cell cycle.
Are you going to post a Kahoot Friday or like could you post it earlier? If I decided to go back to study all this stuff?
Yeah, I can post it earlier. Yeah, because I'm not like giving any bonus points out based on it. I just don't post it until afterwards so people see it for the first time in here, but that doesn't really matter. I can post it earlier. Yeah, I'll make a mental note to myself. Okay. Here we go. Sharing. Oh, no. Okay, come on. Oh my gosh. Okay, so the cell cycle, let's see. We finished talking about all of the parts of a cell, like the cell membrane, all of the organelles inside. So now this chapter goes into basically the life cycle of a cell. So if you imagine one cell, what does it do over the course of its life? How does it reproduce? All that good stuff. So that's what we're going to talk about with this chapter. And if you remember the term or have heard the term mitosis, that's going to be sort of the last half of this lecture is mitosis, just the splitting of a cell. So what is the cell cycle? Basically just, if you break it down, it's an ordered series of events that involves cell growth. So the cell actually does grow in size and then it divides. So cell growth and cell division is the overall cell cycle. So there's more to the cell cycle than just mitosis, which is the division part. A really important part and what cells spend most of their time in is actually not mitosis, it's interphase, which is where the cell is growing, adding nutrition, just overall getting larger in size up to a certain point and getting ready for division. And then this is also where DNA is replicated. So the two main kind of phases or steps or divisions of the cell cycle is interphase and mitosis. I'll talk about what happens in each of those. But in general, interphase involves cell growth, DNA replication, because if you're starting with one cell and you want two cells, each of those cells has to have DNA in it. So we have to replicate that DNA before it can split. And then mitosis is just the process of one cell dividing into two. So that's kind of an outline of what we'll be talking about in this chapter. So why do cells do this? You probably have a decent idea of why cells have to divide. Let's start with single-celled organisms. So bacteria, they are only made of one cell. In order to make more of themselves, to reproduce, they have to divide themselves. So they undergo a really simple reproductive process where they just split themselves in two through a process called binary fission. So reproduction is one purpose of the cell cycle. In multicellular organisms, we also develop from a single cell. So all of you guys at one point were just one single cell. And then you divided many, many, many times in order to become who you are now. So growth and development is a really important aspect of, from a multicellular organism perspective, the cell cycle is really important in growth and development. And you're constantly losing dead cells. So your skin cells are constantly sloughing off, they're dead on top, and then new cells replace those. So in order to regenerate these tissues, you have to make more cells. Otherwise, if you're constantly losing cells and they're not being regenerated, you eventually have no tissue left. So like no skin, which would be bad. So tissue regeneration is another important purpose of the cell cycle. These really deal with multicellular organisms, these last two, the growth and development and tissue regeneration. The reproductive aspect that I'm mentioning here is really just a single-celled organism reproducing itself, making another one of itself. So those are the three main purposes of the cell cycle. And here is a diagram of what a cell does over its lifetime. So we're going to go through each of these phases. I want to point out first, kind of guide you through this. We're going to kind of start here. So we'll imagine this one cell has just split from another cell, right? So we have a new cell. The majority of its life is spent in what we call interphase, so not actually in mitosis at all. A really short portion of a cell's life is that mitosis, which gets all the hype and press. Most of the time, the cell's growing, it's replicating its DNA, it's kind of getting ready to divide. And I'll talk about what happens in each of these stages, but there's 3 stages to interphase. G1, S, and G2. You can think of the G as growth, like 2 growth stages. This will be DNA synthesis, which is why it's the S stage. And then the mitotic phase, or M phase, like I said, takes up a pretty small portion, but we'll talk about it in detail as well. But most of a cell's life cycle, lifetime, is spent in interphase. All right, so that G1 phase, we're going to start down here. We're starting with a cell that just divided off from another cell. So we'll say it's a new cell, pretty new cell. The G1 phase right here is really just for cell growth. That's the main purpose. It's taking in nutrients and growing and developing. The S phase is a really critical phase where DNA is replicated or synthesized. The S comes from DNA synthesis. That's why it's called S phase. But this is where, so in G1, the cell had 1 copy of its DNA. The S phase is where it's going to make another copy. So an exact copy of its DNA. It needs an exact copy of its DNA because eventually it's going to turn into 2 cells down here. So the DNA synthesis stage is where it copies the DNA. So now at the end of the S stage, there's 2 copies of DNA in the cell, 2 exact, ideally exact copies of the DNA. So it's the exact same. And then the G2 phase, I'll talk about some cell cycle checkpoints. This is a big one where the DNA is checked for any errors. So DNA replication is not always perfect. G2 is where that DNA that was copied or made in the S phase is checked, make sure it's okay. If there's major issues, the cell will just die and not actually go on to do anything. But most of the time it's fine, and then it can go into the mitotic phase. And another important aspect of G2, different parts of the cell that are important for cell division, so to actually let the cell divide, are starting to appear, which are centrosomes and centrioles. We won't worry too much about exactly what those are, but they'll come up again when we talk about mitosis. They're basically how the cell kind of gets pulled into, more or less. Okay, so that's interphase. There's a lot going on in interphase, but a lot of it's just kind of, it seems like not a lot's happening because it's growing, it's checking DNA, all that kind of stuff. The S phase is probably the most active part of the interphase piece of a cell cycle. So let's talk about the S phase a little bit more since it's so important. First, we need a little bit of vocabulary, a vocabulary lesson before we get into the specifics of DNA replication as well as mitosis. So some DNA terminology. You know overall that DNA is a nucleic acid. It's one of those main macromolecules that you learned. I don't remember if I've mentioned chromatin before. I don't think I have, but DNA, when it's when the cell's not dividing, it's in this really uncondensed form called chromatin. So it's all like this long stringy stuff, which I'll show you an image of when we get into mitosis. So it's just these long strands of DNA. I think most people have kind of seen this shape of a chromosome before. So this is the condensed form of DNA. If you look at it here, that X shape, that is when the DNA has condensed. So right there. Chromatin is just this long stringy stuff. It doesn't look anything like a chromosome. It's still DNA, it's just different. Chromatin is not condensed, chromosomes are condensed. It's the only difference. When a cell is about to divide, it condenses its DNA down into chromosomes because it just makes it easier. It doesn't get all tangled and mixed up. So chromatin, chromosome, a chromatid, to make it more confusing with a D on the end, is just one double helix of DNA. So I talked about DNA as being this double helix of material. So it looks something like this. So one of those makes up one side of this. That's a chromatid. This gets a little confusing. So this would be a chromatid for you guys on Zoom. Kind of 1/2 of the X. When a cell is ready to divide, ready to go into mitosis, it'll have two of these stuck together, and they're called sister chromatids, and they're exact copies of each other. So when the DNA replicates itself, it's basically replicating 1/2 of this to make the full X. So sister chromatids are exact copies from a DNA perspective of each other. And they're stuck together with a centromere. I think I have that defined on the next slide. Chromatid is just 1/2. Sister chromatids make up the X. So this is essentially what will get pulled apart during mitosis. So each eventual cell has a copy of the DNA. All right, centromeres, like I just mentioned, they're kind of the waist, that little pinched in area of the chromosome. This is where this terminology gets a little bit confusing. So this X shape is a chromosome and just this one chromatid is also a chromosome. So they define one chromosome as a centromere. If there is a centromere present, it's a chromosome. It's really strange. It'll come up again when we go over meiosis, which is a lot further down the road. But this is a chromosome, this one double helix of DNA, or when two of the same double helices of DNA are stuck together, that's also one chromosome. So one centromere indicates one chromosome. That centromere is important because that's where the kinetochore is. The kinetochore is basically an attachment point for the next word on here, the spindle microtubules. So these spindle microtubules, which are just little, basically strands of protein, are going to attach to the kinetochore, also made of protein. And these spindle microtubules are what's going to pull that X apart, pull the chromosome apart into two separate chromatids during mitosis, during the splitting of the cell. So for you guys on Zoom, here's the kinetochore, and then this is the spindle microtubules, which will pull that apart. Okay. So all of those terms will come up again. That's why I at least wanted to bring them up now before I'm going into the details of what happens during mitosis. Any questions? Good so far? Okay. So let's look at, finally, chromosome duplication. So that's the S phase of interphase. So we have, before the DNA is duplicated, or the chromosome, another way to think of it, is duplicated, you just have this one singular chromosome. It gets copied, and we'll talk about exactly how that happens much further down the line in the semester, exactly how this code is copied into another one, but not yet. You can just imagine it magically appears. A new one magically appears next to it. So it exactly duplicates this original chromosome. So it goes from 1 to 2. This is a chromosome. This is also a chromosome. But now you have two copies of the DNA in one chromosome versus just one. That's really like all you need to know for now about DNA replication as far as S phase of interphase. Really, this is just to kind of help you picture what's happening, going from this one stranded chromosome, one chromatid chromosome into the double. So it's just copying the DNA, and just know and burn it into your brain that DNA is replicated in S phase of interphase. Okay, so now let's look at when there's one copy of DNA versus when there's two copies of DNA in the cell. We start out in G1 with our kind of new cell that's just split off. There's only one copy of DNA. So we're starting with one copy of DNA in G1. By the end of S phase, so the whole S phase, the goal is to copy the DNA, right? So now we have two copies of the same DNA at the end of S phase. So that means still going into G2, there's two copies of DNA. And this is where that DNA gets checked. Is there anything wrong with it? No, then it can continue on. Yes, then it can be repaired, or the cell might die. So we have one copy of DNA, and then the next two phases have two copies. And it was hard to squeeze this in, but at the end of mitosis, so this is in where the cell actually splits. So at the very end of mitosis, it's back down to 1 copy of DNA. That's because we've gone from 1 cell to 2 cells. So these 2 cells here. Each of those cells is going to get 1 of these copies. So this is going to go into 1 of the cells, and then this will go into the other cell. So now we're back down to just 1 copy of DNA in the cell. And then we start the cycle all over again. Start with 1 copy of DNA here, synthesize it, etc., over and over and over again. Any questions about this? Okay. At some point, I will ask you, when is there 1 copy of DNA versus when are there 2 copies of DNA? Like when in the cell cycle does that change? All right, moving right along. So we're kind of cruising through this. Am I going too fast? Are you guys good? Okay. Now we're into the super exciting part of the cell cycle, the M phase, or it includes mitosis and cytokinesis. And I'll define what those are in just a second. Okay, so here we are. We're in this little tiny wedge of the cell cycle. Like I said, it doesn't take long for a cell to actually divide into 2. Most of the time is going into growing and synthesizing or copying the DNA. These are the 5 stages of mitosis that you want to know. So we're starting here. It's weird going from right to left, but I think that's because this cell cycle diagram is going in that direction. So we're starting with prophase. I'll talk about exactly what's happening in each of these phases, but this just gives you a good overview. You want to know the order of these different stages of mitosis and then what's happening in each one. So we have prophase first, and then prometaphase, and then metaphase. So prometaphase is in between prophase and metaphase. It's pretty easy to remember. Then you have anaphase, and then telophase, which is followed quickly by cytokinesis. Cytokinesis is where the cell actually splits into. It's not really part of mitosis. It's kind of separated from it, but it's part of the M phase. Don't worry too much about that distinction. So when I learned mitosis, and in some of my classes I still teach it this way, but there was no prometaphase. So prometaphase has kind of been wedged in there in between prophase and metaphase. The easiest way, I mean, it's not really that helpful, but I always remembered PMAT, P-M-A-T, prophase, metaphase, anaphase, telophase. But now there's that prometaphase in there, so it's PPMAT. I don't know if that's, you guys can make up an acronym for that if you want to, but those are the 5 stages you want to know in that order. Okay. So let's go through each of these phases and look at what's happening in the cell. So there we go. That's the order: prophase, prometaphase, metaphase, anaphase, telophase. Okay. What is happening in each of these? In prophase, so before, and I'll show you images on the next slide of these first couple stages. At first, before mitosis really gets started, all of the DNA is in that long uncondensed form, like this long, just kind of a bunch of spaghetti is what it looks like to me in the nucleus. So it's uncondensed chromatin. That's gonna congeal and condense into these nice distinct chromosomes. It'll be a lot easier to split up the DNA in that condensed form. So chromatin condenses into chromosomes. And then these spindle microtubules start to show up. And that's also referred to as the mitotic spindle. You can think of these as sort of the tracks on which these 2 chromatids will separate from each other, like I already described. So these spindle microtubules will basically pull that chromosome apart. So one half of the DNA goes to one end of the cell and the other half of the DNA goes to the other end of the cell. Those are the main processes in prophase. There's other stuff that happens. I've kind of boiled it down to the most important aspects. So if you look at like a textbook or other sources, you might see other events happening in these stages, but these are the ones that I want you guys to be familiar with. I think they're the most critical to know. All right, on to prometaphase. The nuclear envelope is going to break down. So we know the DNA is in the nucleus. In order for that DNA to go to either side of the cell, there can't be a nuclear envelope. So that has to break down in order for the DNA to be able to move out. And now the spindle microtubules are attaching to that kinetochore, which is basically the center of the chromosome. So those tracks are actually connecting to the chromosome, which it will pull apart. So that's prometaphase, not a whole lot happening. Here are some images, because I always think, I don't know, I feel like pictures help. Okay, so we're gonna start, here's interphase. So this is even before we get into mitosis, into the M phase. Here you can see the chromatin. So you don't see any chromosomes, it just looks like a bunch of spaghetti, like I said. So interphase, you know it's interphase when you don't see any specific chromosomes. The DNA hasn't condensed yet. Here you can see the condensed DNA as chromosomes. So here, the chromosomes, back in interphase, it was just this big mess of DNA. Okay, we start to see that mitotic spindle form. So again, these are the tracks that'll pull the DNA to either side of the cell. Prometaphase, the nuclear envelope is breaking down. And now these chromosomes you can see are attaching, or the spindle microtubules are attaching to the chromosomes. So over here, they were kind of on their own. Now they're coming over here and attaching to the chromosomes. So for you guys on Zoom, here's the spindle microtubules attaching to the chromosomes. That's what's going to pull them apart eventually. Okay. So those are the main events that happen in prophase and prometaphase.
Okay, did you say that green thing, the spindle?
Yes, this.
Okay. You said those surround the...
These are going to attach to either side of the chromosome, and then they're going to pull those apart. So it's like a track that each half of that chromosome is going to move on towards either end of the cell. Okay, into metaphase. Metaphase is the easiest one. That's the easiest stage. This is where chromosomes are just lining up along the middle. So in that previous image, like you saw, the chromosomes were kind of all over the cell. Now they're going to all line up along the very middle of the cell. Nice and neat. So metaphase, middle. That's how I remember metaphase. 2 M's. Anaphase, also pretty easy. This is where those sister chromatids that are connected at the centromere are going to pull apart and move to opposite ends of the cell. So far we've basically just been preparing for this, for anaphase, because we want that DNA to move and to separate onto either end of the cell, because that will eventually become 2 cells. So metaphase and anaphase, 2 of the easier ones to remember because there's really only one thing happening. And then telophase, which is the last one, or telophase, however you want to say it. So now you can imagine, and I'll show you an image, you have a copy of DNA on one end of the cell, a copy of DNA on the other end of the cell. Now there's going to be nuclei, 2 nucleuses that are forming around each of those sets of DNA. Now, because the DNA doesn't have to move around anymore, it also just decondenses back into chromatin, into that spaghetti-like mess of DNA. So that's telophase. And then cytokinesis, which is the actual splitting. So you can see from this bullet pointed list, telophase is the last stage technically of mitosis, but we haven't actually made 2 cells yet. Cytokinesis is where that one cell, now with a copy of DNA on either end, is going to split in 2. Animals and plants do it differently, slightly differently. Animals develop what's called a cleavage furrow, and I'll show you what that looks like, but it's basically the big overall cell kind of pinches in 2 from the outside. Plants develop what's called a cell plate, which grows from the middle out, and I'll show you what that looks like. So a cleavage furrow versus a cell plate. You want to know that difference between plants and animal cells as far as cytokinesis goes. Other than that, the whole M phase as well as interphase is pretty much the same in plants and animals. This is kind of the main difference when we're talking about the cell cycle between plants and animals. That very final stage happens slightly differently. So the cells split a little bit differently. And then next, we're going to look at, just like that previous slide, images of each of these things happening. And I'll describe what's happening again in each of these phases, and you guys can see kind of what it looks like. All right, so metaphase, this is where the chromosomes line up along the midline of the cell. It's called the metaphase plate. You don't need to know that, but it's aptly named. So chromosomes are lining up. You can see the spindle microtubules are attached to either side. We move into anaphase, and that's where those spindle microtubules pull those chromosomes apart. So anaphase, the chromosomes are being pulled to either end of the cell. And then telophase and cytokinesis are oftentimes just kind of lumped together. So here we have telophase and cytokinesis kind of happening. It all happens at about the same time. But you have a new nuclear membrane kind of forming around each of these eventual new nuclei. So now you can see all of this DNA is going down here. It's going to be in this cell. All of this DNA on top is going to go over here. It'll be in the other cell. So this DNA will move up. This DNA will move down. And then you have a complete set of DNA in each of these eventual cells. Because remember, each of these are a copy. So those are exact copies of each other, each of those chromatids. All right, and the cleavage furrow is what's being shown here. So the cell kind of pinches in from the outside. That's in an animal. I'll show you what it looks like in a plant cell. Okay. Oh, I forgot I had a video here. It's a really short video with no actual words, I don't think. It just shows mitosis sort of in action in a cell, since it's not a static process. So for you guys, I'm going to hold this up here. So I'm going to kind of narrate it as we go since there's no sound. All right, so you'll see the chromosomes starting to kind of condense. We're looking at all of this. So you can see the chromosomes starting to kind of form. They're going to move towards the middle. Oh, sorry, I can make this bigger. Towards the middle of the cell eventually. Takes them a little while. What's that? Okay, so now you can see I'm kind of moving towards the middle of the cell here. They're about to line up in metaphase. Oh, and there's anaphase. So anaphase happens pretty quickly, but you can see they lined up and then almost immediately get pulled to either end of the cell. So that's anaphase. And then you'll see the cell split in two in cytokinesis. You can't really tell what's happening with telophase there, but so that's what it actually looks like. That's what's happening. It's time lapsed. I don't know how long, I can't remember how long that takes. But that's what's happening in your cells like all the time. It's pretty crazy. Okay. That was mitosis. I think I might almost finish this today. Any questions so far? Okay. Next. Okay, let's look at cytokinesis real quick. So this is how it looks differently in an animal cell versus a plant cell. Like I said, an animal cell, we just saw that happen in that time-lapse video. And the cleavage furrow kind of forms from the outside in. It's like the one cell is being pinched in two, as if you were actually pinching it from the outside. The plant cell, the cell plate, which is developing, starts in the middle and then grows outward.
I have a question.
Yes.
What is that thing, that little board thing in there?
That is the centromere, which is part of that spindle apparatus. So it's kind of like the anchor. So the things that the microtubules that pulled that chromosome apart, they're anchored here basically. Good question. Yeah, those are centromeres. So she was, for you guys on Zoom, she was asking about these T-shaped things. Those are centromeres. Or sorry, centrioles. It doesn't matter. You don't have to know the name necessarily, but centrioles, but that's where those spindle microtubules are pulling, essentially anchored to and pulling the chromosomes apart. I get all those centros confused. Okay, so they're also in plant cells. It's just not showing them here for some reason. But yeah, cell plate forms from the middle out. Cleavage furrow forms from the outside in. That's the main difference. Different material that's making it up also, but don't worry too much about that. So it's really, it's kind of easy to take pictures of plant cells during different stages of mitosis. So this is what all the stages look like under a microscope. So I do want you to be able to identify these images as to what stage they're in mitosis. If I give you an image of a stage of mitosis and ask you what stage of mitosis is this, it'll be a really obvious one. Some of them are kind of in between a couple, but it'll be very clear as long as you know what you're looking for. So in prophase, it kind of looks like a bunch of nothing. You still have a bunch of DNA kind of starting to condense, but it's all still in the same area. Prometaphase, I don't know that these for me are hard to distinguish. You have more condensing of chromosomes, but sometimes that's considered in prophase as well. So I usually don't ask you to identify prophase or prometaphase. So you can kind of make a note of that, because I think they are hard. I mean, here it's easy to distinguish, but sometimes prophase is considered more along this line. Anyway, metaphase, pretty easy to identify that midline lining up. These are still kind of strung out a little bit. They're not like perfectly lined up, but you see that line along the middle. Anaphase, they're starting to separate. And I don't think this is a really great image of anaphase, honestly. There's much better images of anaphase where you see, like in that video, it almost looked like a spider, like being pulled apart. And then telophase, in this plant, you can see the cell plate kind of developing, and you have these two daughter nuclei that have formed. All right. Real quick. Eukaryotes and prokaryotes do this slightly differently because prokaryotes are the single-celled organisms. All of that mitosis stuff I was talking about was with eukaryotes, multi-celled organisms. Prokaryotes are a little, well, they're simpler overall, and they also technically don't undergo what's called mitosis. I mentioned this a while ago in this lecture, kind of towards the beginning, but they undergo what's called binary fission. So you want to know that. Prokaryotes undergo binary fission, not mitosis. The main reason, the main difference is that prokaryotes, our DNA is linear, right? So we have a double helix. It forms into that chromosome shape. Prokaryotes, bacteria, have circular DNA. So their DNA is in a circle. So it doesn't divide in the same way. They basically just replicate one of those circles into two, and then the cell splits. It's a lot simpler. With a single-celled organism, it can be. That replication process starts at what's called the origin of replication. So we didn't go into too much detail on the replication in eukaryotes, but the origin of replication is a pretty important notion as far as bacterial DNA goes. So that's just where the actual copying starts. And then, like I said, it'll split into two individual circular pieces of DNA, and then the cell will split in two. That's really all you need to know about prokaryotic cell division. It's called binary fission. Their DNA is circular, and the replication process starts at the origin of replication. Those are the three main points. The big take-home message is that it's different in prokaryotes versus eukaryotes. Okay, I think I have time to finish this. It's pretty brief. So we'll get through this and then we'll just have all Friday to review. Okay, so the cell cycle is highly regulated. So you may, oh wait, I do have a video. Yeah, I'll keep going for a little bit. I'll show you the beginning of this and then we'll kind of finish it up on Friday. Without regulation of the cell cycle and uninhibited division, that's basically cancer. So cancer cells are ones that don't stop dividing. They just keep dividing. So knowing that most of our cells are highly regulated is really important. There's three main checkpoints, and I'll review these again on Friday, but these are the three checkpoints you'll want to know. If you look up information online, sometimes they add an S phase checkpoint, but don't worry about that. Just know these three checkpoints. The G1 checkpoint is happening in the G1 phase of interphase. So there's two checkpoints that happen in interphase, the G1 and the G2. They happen in those phases. And then there's a checkpoint during mitosis as well. And these checkpoints are checking for different things at different times. So the G1 checkpoint is just kind of making sure the cell's big enough in order to continue on and eventually divide. Does it have enough nutrients? Has it grown enough? Is there any DNA damage? Don't worry about the resting state right now. We'll talk about that on Friday. And then if all of that's good, it'll go through DNA synthesis into the G2 stage. And then the G2 checkpoint will check for cell size. And also, again, was the DNA replicated correctly? Are there any errors in the DNA replication before we make another cell based off of that DNA? If no, then it goes, if everything's good, goes into the mitosis stage. And then the only metaphase checkpoint is basically making sure that those spindle fibers are attached to the chromosomes correctly, so they separate correctly. So that's the metaphase checkpoint. Okay, like I said, I'll go over those again since I did that kind of quickly. We'll watch a video and then do a review on Friday. So that's it.